Okay, we're gonna start. Hello, my name's Stephanie Albin. I'm the Senior Science Program Officer in Neuroscience at the Kavli Foundation, and I'm gonna be moderating our penultimate session on open platforms, AI, and data sharing for multidimensional neurobiology. So you may have noticed this session's a bit different than the others based on the furniture. It's going to be a panel discussion. So we have 3 speakers here. Each person's going to speak for about 10 minutes or so, and then there's time for a couple speaker-specific questions after each talk. I ask if you have broader questions that would be applicable for all of the speakers, please hold those for the discussion at the end. I personally am really excited about this session. I think no matter what your experimental question is, what organism you're studying, what techniques you use, data really is at the heart of discovery. But we know data can be messy and complex and difficult to share. And so we hope that this session will be both inspiring, but also offer some practical advice on how to use open data approaches and talk about data collection, management, and the infrastructure needs of this neurobiology and changing environments community. So our first speaker is Rachel Parkinson. She is a Schmidt AI in Science Fellow at the University of Oxford.
Great, thank you, Stephanie. Okay, can everybody hear me all right? Yeah, excellent. So yeah, thank you very much, Stephanie. I'm really happy to be here today. I'm going to be telling you guys a little bit about a project that I dreamed up about 9 months ago and have been working on with a pretty large group of people over the last, 8 months or so, called MetaBee AI. And I hope it's interesting to all of you and relevant to this session as well. So my research really sits at the interfaces between these three fields. So I've done work on the impacts of pesticides on sensory processing, such as visual motion and taste. And I've done this using insect models like the locust and the honeybee. I've also developed software for analyzing electrophysiology signals, and I'm also currently working on a new package for single sensillum recording analysis, if anyone's interested in that. I also am building this behavior lab, which is an automated high throughput system for detecting sublethal toxicity in insects. And the project that I'm going to tell you about today is this large language model assisted meta-analysis pipeline. So I've been really interested in pesticides since I was an undergraduate, and primarily these neonicotinoid pesticides, which are nicotine mimics. And they were interesting to me because I could use them as sort of a pharmacological tool to probe neural circuits in locusts at that time. They were also interesting to me because I come from the prairies of Canada, which is a really highly agricultural area, and people were talking a lot about neonicotinoid pesticides when I started in research. So I wanted to know, how are they impacting neural circuits in these insect species, both in the ones that we'd rather kill and also the ones that we'd rather don't die. A lot of people, as it turns out, are thinking about this, and you may have come across loads of research already on neonicotinoids and bees, especially honeybees. This is an example of an adverse outcome pathway. So for those of you not familiar with toxicology, this is the way that toxicologists try to put together information that might be measured from many different experiments in order to understand how a molecular initiating event, in this case, like the neonicotinoid pesticide binding to the nicotinic acetylcholine receptor, how it ultimately results in some adverse outcome. So in this case, colony death or failure. And what the yellow boxes denote are really experimental endpoints, so things that we're actually measuring in the lab or in the field when we're doing experiments or collecting data. So things like impaired learning and memory, which could be measured at the level of the neural circuit, or it could be measured through behavior. Foraging activity, there's lots of different types of endpoints in here, and it's actually very challenging to understand where or how these white arrows work and where to put them. We know that this is also a much more complicated situation where we don't just have single pesticides having an impact in an environment, but there's lots of other chemical stressors, especially in an agricultural environment. We also have parasites and pathogens, extreme weather events or climate change, things also like habitat loss or nutritional changes based on habitat change. So it's really complicated. And this is just thinking about honeybees. We've got hundreds or thousands of species of bees in any given area, and these effects will be present in all of them. So my big question here is, what can existing data tell us about how animals are responding to environmental change? And really, how can we use predictive models to guide future research? So how can we take the information that already exists, put it into some kind of computational model that might give us information about research that we should do, like pieces of evidence that we're really missing to fill in something to sort of flesh out the model, and which species potentially we should focus on, or kind of branching out from there. So how can we use these models to guide our research? And that brings me to this project, MetaBee AI. So here we are using large language models to help us speed up various steps involved with meta-analysis. And this pipeline that we're developing, really, I'm hoping will be applicable in lots of different areas. So my specific use case is just one example, but I would love to see this in action in different areas as well. So it really involves two realms here, two lines of effort. Firstly, the expert in the loop large language model pipeline, which is being developed currently for systematic review in biology. And we're really planning to create this very user-friendly and interactive software so people who do not have a ton of coding experience can interact with it and adapt it for their own needs and their own research questions. We are fine-tuning these language models on biological data so that actually they become better at reading papers where there might be a lot of really nuanced terms that are very specific to our different areas. And very important to us is to have this expert in the loop verification, because we all know that large language models, or you might know that large language models make things up sometimes, they hallucinate. So what I mean by large language models, as I'm sure you've all already interacted with them through things like ChatGPT, they're fantastic tools. They work really well for a broad range of tasks, but they can't always be trusted. And when we're doing science, we want to really make sure that the science we're doing is correct. So we need to be able to verify where information is coming from and whether or not it's actually accurate. The other side of the project is involving a lot of bee ecotoxicologists, or people in general who are researching bees. And we're working on this bees and specifically nicotinic cholinergic, so that's the neonicotinoid pesticides and related ones, and to see how we can use the pipeline in order to basically perform a large-scale systematic review in this topic, so using all of the pesticides in this category and all species of bees, and using all of the data that exists in the literature already. And a really cool sort of endpoint goal of this project is to create a living pipeline. Feels like at least once a week I get a message in my Gmail that another Google Scholar alert about a new paper on bees and neonicotinoid pesticides or a related compound. Wouldn't it be wonderful if these data could be incorporated in models as we go along? So it's not just that we do a meta-analysis every 5 years, but actually that it's updating. So the data that we're collecting is then actually going to contribute to a larger model. So what does this pipeline actually look like? I'll walk you through a little bit what we've done so far and what we are still working on. So the first step, of course, is to acquire papers, and I did this in the usual way, where I searched on Scopus and Web of Science for some very specific terms. I then screened those initial papers, very large volume of papers, the abstracts and titles for relevance. The next step was to convert all of those PDFs to text, which seems as though it should be a very simple step, but actually we ended up using an AI model for this step as well, which worked out really, really nicely. And if you're interested, I can tell you all about that at a later moment. The next step is, of course, the pipeline. So this is the part where we are querying the text data from individual papers. We're looking at one paper at a time because we want specific information from each and every single one of these papers, and getting things like experimental metadata. So what was the experiment that was done? What was the pesticide that was used? What concentrations? What species of bees? There's all this information. And then the more specific information, like the results. So what were the variables that were being measured? What was the mean and the standard deviation? Numbers that we can actually use in a meta-analysis. One thing that large language models are really good at is summarizing text. So something that we've been harnessing quite a lot is getting text summaries from the paper. So things like, was the overall effect significant? Or what did the authors recommend for future research directions? Did they identify any pitfalls or limitations in their study? Things that we can use to then kind of consider what we might be doing to improve the field. So what really should we do next? Kind of a hypothesis generation step. And then, of course, we have our expert in the loop output review. So we've built some software in order to facilitate this process. The final stage is then, of course, to database all of this data, so to keep the metadata from the papers alongside the actual sort of numerical measurements that we're taking. And then we can do a whole bunch of different type of analyses. So we're not actually at this step yet, but we're on our way to get there. So this is a little glimpse of what our software looks like. It's by no means beautiful, but one very cool thing that it does is that when you look at a particular question that the large language model has been asked about the paper, it gives you the specific piece of text where it came from. So as a reviewer, we can then look at the answer. So here's the AI answer. Bumblebees, specifically Bombus terrestris and Bombus terrestris dalmatianus, were tested. So these are all the same word. And we can see why the model came to that conclusion and where it got the answer from. So then our reviewers can go in and say, okay, yes, this was a good answer, or this is what the answer should have been, and give their opinions on that. So our initial data set, as I mentioned, it contained about 4,500 papers, which I whittled down to just over 1,000 papers. So for the context of this project, we weren't including things like papers that were focused exclusively on residue measurements in the field. We really wanted papers that were going to be describing effects of these specific pesticides or any combination of these pesticides with any other stressor on a species of bee, any species of bee. So we ended up with just over 1,000 papers. Then using a combination of both the large language model and then also at the same time having our experts review the papers, we whittled these endpoints, so specific measurements that were made from the papers, down into these categories, or what we're calling biological domains. So molecular being things that are measured at the level of the molecule, like enzymes or gene transcription. Sub-individual being anything that's measured by cutting up an animal, so effects on, let's say, the brain or the gut. Individual measurements, which tend to be primarily behavioral. Population, which could be anything at the level of the colony or the hive. Community, which is looking at interactions between different species. And then we have a special category for the LD50 measurements. These are the typical gold standard toxicology endpoint, the lethal dose that kills 50% of the population, which is in fact a population measurement, but we're keeping it separate for the reason that it's this very important measurement. And so we look across all of our papers, all of the endpoints that were measured. We see that there's fairly even distribution of most of the biological domains, except, of course, the community domain, which these are very difficult experiments to run, where you're trying to look at interactions between species, or even just measuring more than one species in the wild at the same time, where there are very few papers. We also found that while many of the papers only focus on a single biological domain, so a single one of these categories, several papers, or many papers, also look at more than one domain, and I can break down things like correlations between domains, but we don't have time for that right now. I guess my reason for showing you all this is that there's really many different types of endpoints across various levels of biological organization, and a key challenge that we're facing is to try to define what the hidden variable could be. So when you have a measurement that's something like changes in firing rate or adaptation rates, what we're really looking at is neural circuit function. And so we want to be able to teach our models how to define these hidden variables so we can more quickly categorize these endpoints. And because I've been told that word clouds are the pie charts of 2020s, I just very quickly generated these two word clouds from all of the data that we have. So unsurprisingly to anybody who studies these pesticides, imidacloprid is the most represented pesticide across all the papers, and the honeybee is the most represented bee. But this is the kind of stuff we can do very quickly and easily with that text data that I was telling you about. So if we come back to our pipeline, we are currently working on a benchmarking data set. So I have about 18 biology experts, pollinator ecotoxicology experts who are using the software to generate this benchmarking data set. We are refining the pipeline so to make it work better both in our context and hopefully more broadly for biology. And we are continuing with the software and tutorial development. With the database and analysis side, we're working on the analyses, of course, at this time of those text outputs. We're also interested in finding new ways of obtaining the numerical data, both from the supplemental information attached to the papers, as well as from the figures themselves. And we will be using these data for various modeling techniques. So we had a workshop just a couple of weeks ago in Oxford, which was really wonderful. Here's a big list of all the collaborators on the project. As you can see, there are lots of people. A subset of those people, about 20 of them, came to Oxford. 15 of those people are ecotoxicology experts, and about 5 or 6 of them were AI experts. And we spent a lot of the workshop talking to each other about how to communicate better between disciplines. We had hands-on large language model workshops, for example, for the biologists, which I think they found really, really valuable. Thinking about interdisciplinary science, I mean, these are 3 of the people that I've been working most closely with on this project. So Shusheng Cao, who helped me develop the large language model pipeline, is a quantum physicist. Mikhail Miskolainen is a particle physicist, so dealing with the software side. I do have one biologist here, but he's actually a computer scientist by background. So yeah, finding ways to communicate with people is really important, but also I think what it's really shown us is that it's very helpful to have a domain expert leading a project like this who can kind of make these communications between people a little bit more feasible. So it's not an easy job, but I guess somebody's got to do it. So thank you all very much. Please chat with me if you would like to hear more or collaborate on a project or see how you could adapt this for your own purposes. And also, I will be starting a lab very soon at Queen Mary University in London, so if you have any reason to want to move to the UK, that might be possible. There are some postdoc-specific fellowships that include covering visa costs and all of that. So yeah, very happy to chat more. Thank you.
Okay, we have time for a couple questions.
How many MS can I have so many questions. I mean, obviously, it's an open field, but I think 2 things. 1, are you making us obsolete? 2, in a way, this creates like a memory or like what used to be review papers every couple of years. Now you're sort of like putting this all into an AI or a large language model, right? That you can question at any point in time. But my big question is, are you envisioning this to be like a subject-specific model that you have multiple of those. Say, for example, one for bees, or specifically for those nicotinoids or so, or one for SDG, and will that then become eventually, if you spin this like into the future, like down the road, will then this large language model be like an editor in a journal where you're like, okay, I have some new data or some new paper that I want to publish, and then this AI will be there and be like, this sounds good, because we don't have this data yet, or it will be like, no, this has been done before, so you're out of luck. Just thinking about like long term, where you would want to see this.
Okay.
But my real question is, do you think this is
Okay.
do you think it's like separate ones?
Yeah, okay. So I think I kind of understand your train of questions. I do not think that we are going to become obsolete anytime soon. I do think that AI is going to help us to speed up the tasks that are repetitive and mundane, and also potentially help us to draw connections between things that maybe we wouldn't have considered previously. So I see this as a help, not as a threat. Where to go next? So in terms of whether or not I think that this is going to be something very specific within individual domains, no, absolutely not. So the really wonderful thing that we've already seen from large language models is just by being trained on loads of data, they're able to generalize quite a bit across different topics. In biology, we're using a lot of similar terminology or similar concepts all the time, so some fine-tuning in this particular area will be really valuable for everyone across all of the individual subsections of sort of biosciences, and specifically things like working with weird animals that we might not typically encounter this terminology. And actually I ran into this problem a lot when I was developing the prompts for the pipeline, where I kept using terminology that to me seemed so obvious, like the word endpoint. And then people around me, like the physicists, and actually some of the biologists also were like, what do you mean by endpoint? Like, what is this? To me, that's like this, or that's like this thing. And I'm like, oh, maybe this is a toxicology term. And maybe I think it's like a broad term, but it's kind of not. So actually, this will be good for everyone. I can't remember what else you said, but I feel like don't be afraid. We'll be okay.
Hi, Rachel, really cool thing that you're doing, but I found myself wondering, you're using published papers that have been through peer review. Do you include things like bioRxiv, things that have not been through peer review, or even going further, unpublished data that were not significant? Are there ways of encouraging people to make those data sets available? Because it strikes me that would be very informative for what you're trying to do.
Yeah, I think that this is hitting it on the head. It's a real shame how many data sets exist that aren't published because they didn't find anything cool enough to publish, and negative results are really important. So absolutely we can use things off of the archive, also including gray literature, so things that are published by government or even by pesticide companies could be really valuable, but again, keeping a tag on where the different data types are coming from so that we can, as experts, question them if need be. Thank you.
So it strikes me that there could be several confounds in what the LLMs are doing. So for example, the word neuromodulation is used completely differently by different subsets in our community. And I'm sure if you look at things like adaptation, there'll be 12 different definitions. And so I'm a little bit confused about how whatever it's doing is going to untangle the context of those ambiguous terms.
Yeah, thank you that's a great question. It's, I think individual words can be very confusing, but when you put them into an appropriate context, the meaning becomes more obvious. And it's really challenging to do this when we're just having a conversation with somebody who's embedded in their own discipline and we have a mismatch in how we're using a word. But the idea behind fine-tuning these language models so they become better at understanding the contexts in which words are being used, we would hope that then, if you're using it in a context where you are talking about, the stomatogastric ganglion in a crab, then it will know what you mean because of that context. So I don't know if I can fully answer your question. I'm sure that Ben or Saskia could probably give a better explanation on this, but that's sort of my impression of how these things work. It's about context rather than just the individual word.
Okay, we have time for one last quick question.
I'm curious if you might envision this as a mechanism for automating the generation of AOPs. So I could imagine as maybe an exercise, you go to Dioxin, you get rid of all the AOP papers, and you just interrogate the data papers and see if you could train it to build an AOP and how well it would do.
I love that idea. Adverse outcome pathway. So the thing that I showed with the B and all the different endpoints, and yeah, because these are really, really used in toxicology. People care about this stuff a lot. Yeah, that's a great idea. Wonderful. Haven't tried it, but yes.